Welcome to Urgene International, Internal Auditor course for the requirements for quality and competence for medical laboratories based on ISO 15189, 2022. Process requirements. The laboratory must identify any potential risks that could affect patient care across all stages of testing, from pre-examination, through the examination itself, to post-examination processes. Once these risks are identified, they must be carefully assessed and reduced as much as possible. If any risk remains, the laboratory should communicate this residual risk to users when appropriate. The lab must also keep track of these risks over time and evaluate how effective the mitigation measures are, based on the level of potential harm to patients. In addition to managing risks, the laboratory is responsible for identifying opportunities to improve patient care. It should establish a clear framework for managing and acting on these opportunities, as referenced in Section 8.5. The laboratory must have well-defined procedures for all pre-examination activities, and these procedures must be easily accessible to all staff involved. Pre-examination steps, such as patient preparation, sample collection, handling, and transport, can significantly affect the quality and reliability of the final test results. For detailed guidance, ISO 20658 outlines requirements for sample collection and transport. Additional standards provide more specific information for certain sample types and analytes, including ISO 20186-1, ISO 20186-2, ISO 20186-3, ISO 20166, all parts, ISO 20184, all parts, ISO 23118, and ISO 4307. These references help ensure that pre-examination processes are carried out correctly and consistently, supporting accurate and reliable patient results. Laboratory information for patients and users. The laboratory shall ensure that appropriate information is made available to its users and patients. This information shall be sufficiently detailed to provide a comprehensive understanding of the laboratory's scope of activities and requirements. As appropriate, the information shall include the laboratory's location or locations, operating hours, and contact details, as well as the procedures for requesting examinations and for the collection of samples. The information provided shall also include the scope of laboratory activities and the expected timeframes for the availability of results. It shall describe the availability of advisory services and outline any requirements for patient consent. Additionally, the information shall specify factors known to significantly affect the performance of examinations or the interpretation of results, and it shall clearly set out the laboratory's complaint process. This ensures transparency, accountability, and accessibility for both patients and users. Requests for providing laboratory examinations. Each request accepted by the laboratory for examinations shall be regarded as an agreement. The examination request shall include sufficient information to ensure the following: unequivocal traceability of the patient to both the request and the corresponding sample. Clear identity and contact information of the requester. Precise identification of the examinations being requested. The examination request shall also include sufficient information to ensure that informed clinical and technical advice, as well as clinical interpretation, can be provided. The request information may be submitted in a format or medium deemed appropriate by the laboratory and acceptable to the user. Where necessary for patient care, the laboratory shall communicate directly with users or their representatives to clarify the details of the request, ensuring accuracy and completeness. Oral requests. Where applicable, the laboratory shall establish a procedure for managing oral requests for examinations. This procedure shall include the requirement for documented confirmation of the examination request to be provided to the laboratory within a specified time frame. Such measures ensure clarity, accountability, and traceability in the request process. Primary sample collection and handling. The laboratory shall establish and implement procedures for the collection and handling of primary samples. Information on these procedures shall be readily available to all personnel responsible for sample collection. Any deviation from established collection procedures shall be clearly documented. The associated risks and the potential impact on patient outcomes, whether from acceptance or rejection of the sample, shall be assessed, recorded, and communicated to the appropriate personnel. The laboratory shall also conduct periodic reviews of requirements for sample volume, collection devices, and preservatives for all relevant sample types. These reviews shall ensure that neither insufficient nor excessive amounts of sample are collected, and that all samples are properly preserved to maintain the integrity of the analyte. Information for pre-collection activities. The laboratory shall provide clear information and instructions for pre-collection activities, with sufficient detail to ensure that the integrity of the sample is preserved. This shall include guidance on the preparation of the patient, including instructions for caregivers, sample collectors, and patients. It shall also include the type and amount of the primary sample to be collected, descriptions of the required containers and any necessary additives, and, where relevant, the correct order of sample collection. Before collecting any samples, certain key information must be clearly communicated. This includes any special timing requirements for when the sample should be collected, as well as any clinical details that may influence how the sample is collected, how the test is performed, or how the results are interpreted, for example, whether the patient has recently taken any medications. Each sample must also be labeled in a way that leaves no room for confusion. Labels should clearly identify the patient, specify the source and site of the sample, and distinguish between multiple samples taken from the same patient, such as several tissue pieces or slides. Additionally, the laboratory must define its criteria for accepting or rejecting samples based on the specific tests requested. This ensures that only suitable samples proceed for examination, maintaining accuracy and reliability in reporting. Patient consent. The laboratory shall obtain the informed consent of the patient for all procedures performed on the patient. Note: for most routine laboratory procedures, consent may be inferred when the patient willingly submits to the sample collection process, such as venipuncture. Special procedures, including those that are more invasive or carry a higher risk of complications, may require a more detailed explanation and, in certain cases, documented consent. In emergency situations where obtaining consent is not possible, the laboratory may proceed with the necessary procedures, provided that such actions are taken in the best interest of the patient. Instructions for collection activities. To ensure safe, accurate, and clinically appropriate sample collection and pre-examination storage, the laboratory shall provide clear instructions covering the following: verification of the identity of the patient from whom the primary sample is collected. Verification, and when relevant, documentation that the patient meets the required pre-examination conditions, such as fasting status, medication status including the timing of the last dose or cessation, or collection of the sample at a predetermined time or interval. Procedures for the collection of primary samples, including descriptions of the required sample containers, any necessary additives, and, where relevant, the correct order of collection. The labeling of primary samples in a manner that provides an unequivocal link to the patients from whom they are collected. The recording of the identity of the individual who collected the primary sample, together with the collection date, and, where relevant, the collection time. The requirements for separating or dividing the primary sample, when necessary. The stabilization and appropriate storage conditions of collected samples prior to their delivery to the laboratory. The safe disposal of all materials used in the collection process. Sample transportation. To ensure the timely and safe transportation of samples, the laboratory shall provide clear instructions covering the proper packaging of samples for transportation, ensuring that the time between collection and receipt by the laboratory is appropriate for the examinations requested, maintaining the specified temperature conditions required for sample collection and handling, any specific measures necessary to preserve sample integrity, such as the use of designated preservatives. If the integrity of a sample has been compromised and poses a health risk, the organization responsible for transporting the sample shall be notified immediately. Appropriate action shall be taken both to reduce the risk and to prevent recurrence. The laboratory shall establish and periodically evaluate the adequacy of its sample transportation systems to ensure reliable and safe handling. Sample receipt. Sample receipt procedure. The laboratory shall establish and implement a procedure for the receipt of samples. This procedure shall include ensuring unequivocal traceability of each sample, through both the request and the labeling, to a uniquely identified patient, and, where applicable, to the anatomical site. It shall also include defined criteria for the acceptance and rejection of samples, and, where relevant, the recording of the date and time of sample receipt. The recording of the identity of the person receiving the sample, where relevant. The evaluation of received samples by authorized personnel to ensure compliance with the established acceptability criteria for the requested examinations. Instructions for the handling of samples specifically marked as urgent, including requirements for special labeling, transport conditions, rapid processing methods, defined turnaround times, and special reporting criteria. Ensuring that all portions or aliquots of a sample remain unequivocally traceable to the original sample. Sample acceptance exceptions. The laboratory shall establish a process that prioritizes the best interests of the patient in cases where a sample has been compromised. Such compromises may include incorrect patient or sample identification, sample instability due to delays in transport, improper storage or handling temperatures, the use of inappropriate containers, or insufficient sample volume. When a compromised sample that is clinically critical or irreplaceable must be accepted, the risk to patient safety shall be carefully considered. In such cases, the final report shall clearly indicate the nature of the problem, and, where applicable, advise caution in the interpretation of results that may be affected. Pre-examination handling, preparation, and storage. Sample security and integrity. The laboratory shall have established procedures and provide appropriate facilities to secure patient samples. These measures shall ensure the integrity of samples and prevent loss or damage during handling, preparation, and storage. Criteria for additional examination requests. The laboratory shall establish procedures that define clear time limits for requesting additional examinations on the same sample. These measures ensure that requests are clinically appropriate and that the integrity of the sample is maintained. Sample stability. Considering the stability of the analyte in the primary sample, the laboratory shall specify and, where relevant, monitor the time interval between sample collection and the performance of the examination. This ensures the validity and reliability of results. Examination processes. The laboratory shall select and apply examination methods that have been validated for their intended use, to ensure the clinical accuracy and reliability of patient testing. Note: preferred methods include those specified in the instructions for use of in vitro diagnostic medical devices, as well as methods published in authoritative textbooks, peer-reviewed journals, international or national consensus standards or guidelines, or in national and regional regulations. The performance specifications for each examination method shall be directly related to its intended use and to its potential impact on patient care. All procedures and supporting documentation, including instructions, standards, manuals, and reference data relevant to laboratory activities, shall be kept current and readily accessible to personnel. Personnel shall strictly adhere to established procedures. The identity of individuals performing significant activities within examination processes, including point-of-care testing operators, shall be clearly recorded. Authorized personnel shall periodically evaluate the examination methods employed by the laboratory, ensuring that they remain clinically appropriate and aligned with the requests received. The laboratory must have a procedure in place to verify any examination method before it is put into use. This verification ensures that the method performs as expected and meets the performance claims made by the manufacturer or described in the method itself. The performance characteristics confirmed during verification must be directly relevant to how the test results will be used in clinical practice. The scope and depth of the verification must be sufficient to ensure that the examination method produces valid, reliable results that support accurate clinical decision-making. Once the verification process is complete, authorized and competent personnel must review the results and document whether the method meets all required performance criteria. If a method is revised by the issuing body, the laboratory shall repeat the verification process to the extent necessary to ensure continued compliance and reliability. The laboratory shall retain comprehensive records of the verification process, including the performance specifications that were to be achieved, the results obtained during verification, and a formal statement indicating whether the performance specifications were met, and, if not, the corrective actions taken. Validation of examination methods. The laboratory shall perform validation for examination methods derived from the following sources: laboratory designed or developed methods, methods used outside their originally intended scope, such as those performed outside the manufacturer's instructions for use, outside the original validated measurement range, or using third-party reagents on instruments other than those specified, where no prior validation data are available. Validated methods that have been subsequently modified. The validation shall be as extensive as necessary to confirm, through objective evidence in the form of performance specifications, that the specific requirements for the intended use of the examination have been met. The laboratory shall ensure that the scope of validation is sufficient to guarantee the validity of results relevant to clinical decision-making. Personnel with appropriate authorization and competence shall review the validation results and formally record whether the outcomes meet the specified requirements. When changes are proposed to a previously validated examination method, the laboratory shall review the clinical impact of these changes and determine whether the modified method may be implemented. The laboratory shall retain comprehensive records of the validation process, including the validation procedure used, the specific requirements for the intended use, the determination of the method's performance specifications, the results obtained, and a formal statement on the validity of the method, detailing its fitness for the intended use. The laboratory must evaluate and maintain measurement uncertainty for all measured values where it is relevant to the test's intended use. This uncertainty must be reviewed against the required performance specifications and properly documented. ISO/TS 20914 provides detailed guidance and examples to support these activities. Measurement uncertainty assessments must be reviewed regularly to ensure they remain accurate and up to date. If there are examination procedures for which evaluating measurement uncertainty is not possible or not relevant, the laboratory must clearly document the reasoning for excluding them from uncertainty estimation. Additionally, information about measurement uncertainty must be made available to laboratory users whenever they request it. If you'd like, I can combine. When laboratory users inquire about measurement uncertainty, the response shall consider all relevant sources of uncertainty, including, but not limited to, biological variation. If a qualitative examination result is based on a test producing quantitative output and is interpreted as positive or negative according to a threshold, the measurement uncertainty of the output shall be estimated using representative positive and negative samples. For examinations yielding qualitative results, measurement uncertainty in intermediate measurement steps or internal quality control results that produce quantitative data shall also be considered for critical, high-risk parts of the process. Measurement uncertainty shall be taken into account when performing method verification or validation, where relevant. Biological reference intervals and clinical decision limits. The laboratory shall define and communicate biological reference intervals and clinical decision limits, where required, to support accurate interpretation of examination results. Biological reference intervals and clinical decision limits shall be clearly defined, and the rationale for their selection documented. They shall reflect the characteristics of the patient population served by the laboratory, while considering potential risks to patients. Note: biological reference values provided by the manufacturer may be used if the laboratory has verified that the population base is appropriate and acceptable for its patient population. Biological reference intervals and clinical decision limits shall be periodically reviewed, and any updates or changes shall be communicated to users. When modifications are made to an examination or pre-examination method, the laboratory shall assess the impact of these changes on associated biological reference intervals and clinical decision limits, and communicate relevant updates to users, as appropriate. For examinations that determine the presence or absence of a characteristic, such as in genetic testing, the biological reference interval is defined by the characteristic itself. Documentation of examination procedures. The laboratory shall document its examination procedures to the extent necessary to ensure the consistent application of activities and the validity of results. Procedures shall be written in a language understood by laboratory personnel and made available at appropriate locations. Any abbreviated versions of procedure content shall accurately correspond to the full procedure. Note: working instructions, flow process diagrams, or similar systems that summarize key information may be used as quick references at the workbench, provided that the full procedure remains accessible for reference and that summarized information is updated concurrently with the full procedure. Information provided in product instructions for use, when containing sufficient detail, may be incorporated into laboratory procedures by reference. When the laboratory implements a validated change to an examination procedure that could affect the interpretation of results, the potential implications of this change shall be communicated to users. All documents associated with the examination process shall be maintained under a formal document control system to ensure accuracy, currency, and traceability. Ensuring the validity of examination results. The laboratory shall establish a procedure for monitoring the validity of examination results. Data shall be recorded in a manner that allows detection of trends and shifts, and, where practicable, statistical techniques shall be applied to support this review. Monitoring activities shall be planned and periodically reviewed to ensure their effectiveness. Internal quality control. The laboratory shall establish and implement an internal quality control (IQC) procedure to monitor the ongoing validity of examination results, in accordance with specified criteria. This procedure shall verify that the intended quality is achieved and maintained, ensuring results remain valid for clinical decision-making. The intended clinical application of the examination shall be considered, as performance specifications for the same measure and may differ across clinical settings. The IQC procedure shall also enable detection of lot-to-lot variation in reagents, calibrators, or both. To facilitate this, the laboratory shall avoid changing the lot of IQC material on the same day or run as a reagent or calibrator lot change. The use of third-party IQC material shall be considered, either as an alternative to, or in addition to, control material supplied by the reagent or instrument manufacturer. Note: monitoring the consistency of interpretations and professional opinions can be achieved through regular peer review of examination results. The laboratory shall select internal quality control (IQC) material that is fit for its intended purpose. When making this selection, the following factors shall be considered: the stability of the material with respect to the properties of interest. That the matrix of the IQC material closely resembles that of patient samples. That the IQC material reacts to the examination method as similarly as possible to patient samples. That the IQC material provides a clinically relevant challenge to the examination method, includes concentration levels at or near clinical decision limits, and, where feasible, covers the measurement range of the examination method. In cases where appropriate IQC material is not available, the laboratory shall consider alternative methods to ensure internal quality control. Examples of such alternative methods include trend analysis of patient results, such as moving averages, the proportion of samples with results above or below specified values, or results associated with particular diagnoses. Comparison of patient sample results on a defined schedule with results obtained using an alternative validated procedure, where calibration is metrologically traceable to the same or higher order references, as specified in ISO 17511. Retesting of retained patient samples to monitor consistency and performance. Internal quality control shall be performed at a frequency determined by the stability and robustness of the examination method, as well as the potential risk of harm to the patient from an erroneous result. IQC results shall be recorded in a manner that allows detection of trends and shifts, and, where applicable, statistical techniques shall be applied to facilitate data review. IQC data shall be reviewed against defined acceptability criteria at regular intervals, and within a timeframe that provides a meaningful assessment of current performance. The laboratory must ensure that patient results are not released if internal quality control, IQC, fails to meet the defined acceptability criteria. If IQC results fall outside the acceptable limits and suggest that patient results may contain clinically significant errors, those results must be rejected. The affected patient samples must be retested once the issue has been identified and corrected, as outlined in Section 7.5. Additionally, all patient samples that were processed after the last successful IQC event must be reviewed to determine whether their results may have been affected. External Quality Assessment, EQA. The laboratory shall monitor the performance of its examination methods by comparing results with those of other laboratories. This includes participation in appropriate external quality assessment, EQA, programs for examinations and interpretation of examination results, including point-of-care testing, POCT, methods. The laboratory shall establish a procedure for EQA enrollment, participation, and performance monitoring for all examination methods used, where such programs are available. EQA samples shall be processed by personnel who routinely perform the pre-examination, examination, and post-examination procedures. External Quality Assessment, EQA. The EQA programs selected by the laboratory shall, to the extent possible, verify the performance of pre-examination, examination and post-examination processes. Provide samples that simulate patient specimens and present clinically relevant challenges. Meet the requirements of ISO IEC 17043. When selecting EQA programs, the laboratory shall consider the type of target value provided. Target values may be independently determined using a reference method, established based on overall consensus data, derived from method-specific peer group consensus data, or determined by a panel of experts. When method-independent target values are not available, the laboratory may rely on consensus values to help determine whether any deviations in results are due to the laboratory's performance or are specific to the examination method being used. In situations where EQA materials are not fully commutable and this limits the ability to compare different methods, it can still be valuable to compare results between the methods for which the materials are commutable. This is often more meaningful than relying solely on comparisons made within a single method. If an external quality assessment, EQA, program is unavailable or is deemed unsuitable, the laboratory must use alternative approaches to monitor the performance of its examination methods. The laboratory must also document and justify why the alternative was chosen and provide evidence showing that the approach is effective. Acceptable alternatives for monitoring examination method performance, when a formal EQA program is unavailable or unsuitable, include participation in sample exchanges with other laboratories, interlaboratory comparisons using identical IQC materials, which allow evaluation of individual laboratory IQC results against pooled results from participants using the same material. Analysis of a different lot number of the manufacturer's end-user calibrator or trueness control material. Analysis of microbiological organisms using split or blind testing of the same sample by at least 2 personnel, or on at least 2 analyzers, or by at least 2 methods. Analysis of reference materials considered to be commutable with patient samples. Analysis of patient samples derived from clinical correlation studies. Analysis of materials obtained from cell and tissue repositories. EQA data shall be reviewed at defined regular intervals against specified acceptability criteria, within a timeframe that provides a meaningful assessment of current laboratory performance. When EQA results fall outside established acceptability criteria, the laboratory shall take appropriate corrective actions, including assessing whether the nonconformance has a clinically significant impact on patient samples. If the impact is deemed clinically significant, the laboratory shall review all patient results that may have been affected and determine whether amendments are required. Users shall be informed of any necessary actions as appropriate. When different methods, equipment, or examination sites are involved, the laboratory shall establish a documented procedure to ensure comparability of examination results for patient samples across clinically significant intervals. Note: the use of patient samples is preferred when comparing examination methods, as this avoids issues associated with the limited commutability of IQC materials. Where patient samples are not available or are impractical, alternative approaches described for IQC and EQA shall be considered. The laboratory shall record the results of comparability studies performed and assess their acceptability. The laboratory shall conduct periodic reviews of comparability of results. Where differences are identified, the laboratory shall evaluate the impact of those differences on biological reference intervals and clinical decision limits, and take appropriate action. The laboratory shall inform users of any clinically significant differences observed in comparability of results. Post-examination processes. Reporting of results. Examination results shall be reported in an accurate, clear, and unambiguous manner, fully aligned with the specific instructions of the examination procedure. Each report shall contain all information necessary for proper interpretation of the results. The laboratory shall maintain a procedure to notify users when examination results are delayed, with the level of urgency determined by the potential impact of the delay on the patient. All information related to issued reports shall be retained in accordance with the requirements of the laboratory's management system. Note: for the purposes of this document, reports may be issued as hard copies or electronically, provided all requirements are met. When users raise inquiries regarding measurement uncertainty, the laboratory shall ensure that its response takes into account other potential sources of uncertainty, including, but not limited to, biological variation. When the qualitative result of an examination is derived from a test that produces quantitative output data and is reported as positive or negative based on a defined threshold, the laboratory shall estimate the measurement uncertainty of the output quantity using representative positive and negative samples. For examinations yielding qualitative results, the laboratory shall also consider the measurement uncertainty associated with intermediate measurement steps or IQC results that generate quantitative data, particularly in critical or high-risk components of the process. Measurement uncertainty shall be taken into consideration when undertaking verification or validation of a method, where relevant. Result review and release. All examination results shall be reviewed and authorized prior to release. The laboratory shall ensure that authorized personnel review examination results and evaluate them in relation to internal quality control, IQC, findings, and, where appropriate, relevant clinical information and previous examination results. The laboratory shall define and implement responsibilities and procedures governing the release of examination results, including specification of by whom the results are authorized and to whom the results are reported. Critical result reporting. When examination results fall within established critical decision limits, the laboratory shall notify the user or other authorized person as soon as relevant, based on the clinical information available. Document all actions taken, including the date, time, responsible person, person notified, results conveyed, verification of the accuracy of communication, and any difficulties encountered during notification. Maintain an escalation procedure for laboratory personnel to follow when the responsible person cannot be contacted. Special considerations for results. When agreed with the user, results may be reported in a simplified format. However, any information required under that is not included in the report shall remain readily available. When results are issued as a preliminary report, a final report shall always be forwarded to the user. Records shall be maintained for all results communicated orally, including details verifying the accuracy of the communication in accordance. Such orally communicated results shall always be followed by an official report. Certain examination results, particularly those with serious implications for the patient, for example genetic findings or results for specific infectious diseases, may require special counseling. Laboratory management shall ensure that such results are not communicated directly to the patient without providing the opportunity for adequate counseling. Anonymized results of laboratory examinations may be utilized for purposes such as epidemiology, demography, or other statistical analysis. Such use shall be undertaken only when all risks to patient privacy and confidentiality are effectively mitigated, and always in compliance with applicable legal and/or regulatory requirements. Automated selection, review, release, and reporting of results. When the laboratory implements a system for automated selection, review, release, and reporting of results, it shall establish a procedure to ensure that the criteria for automated selection, review, and release are specified, approved, readily available, and understood by personnel responsible for authorizing the release of results. The criteria are validated and approved before use, regularly reviewed, and verified following any changes to the reporting system that could affect their proper functioning or place patient care at risk. Results selected by the automated reporting system for manual review are clearly identifiable, and where appropriate, the date and time of selection and review, as well as the identity of the reviewer, are retrievable. Rapid suspension of automated selection, review, release, and reporting is applied when necessary. Requirements for reports. Each report shall include the following information, unless the laboratory has documented reasons for omitting specific items: unique patient identification, the date of primary sample collection, and the date of report issue, displayed on each page of the report. Identification of the laboratory issuing the report. The name or other unique identifier of the user. The type of primary sample and any specific information necessary to describe the sample, for example, source, site of specimen, macroscopic description. Clear and unambiguous identification of the examinations performed. Identification of the examination method used, where relevant. This shall include, where possible and necessary, harmonized electronic identification of the measurand and the measurement principle. Note: logical observation identifiers names and codes, LOINC, nomenclature for properties and units, NPU, NGC, and SNOMEDCT are examples of electronic identification systems. Examination results, with appropriate units of measurement, reported in SI units, units traceable to SI units, or other applicable units. Biological reference intervals, clinical decision limits, likelihood ratios, or diagrams nomograms supporting clinical decision limits, as necessary. Requirements for reports, continued. Each report shall also include, where applicable, lists or tables of biological reference intervals, which may be distributed to laboratory users, identification of examinations undertaken as part of a research or development program for which no specific claims on measurement performance are available. Identification of the persons reviewing the results and authorizing the release of the report. If not included in the report itself, this information shall be readily available when needed. Identification of any results that are preliminary. Indications of any critical results. A unique identification ensuring that all components are recognized as part of a complete report, along with a clear indication of the end, for example, page number of total pages. Additional information for reports. Where necessary to support patient care, the report shall include the time of primary sample collection. The time of report release shall be available when required. If not directly included in the report, it shall be retrievable without delay. The report shall identify all examinations, or parts thereof, that were performed by a referral laboratory. Such information, including details provided by consultants, shall be presented without alteration. The name of the referral laboratory responsible for performing the examinations shall be clearly stated. Interpretation and comments in reports. When applicable, a report shall include interpretation of results and comments addressing the quality and suitability of the sample, particularly where these may compromise the clinical value of the examination results. Discrepancies identified when examinations are performed by different procedures, for example POCT, or in different locations. Potential risks of misinterpretation when different units of measurement are in use regionally or nationally. Observable result trends or significant changes over time. Amendments to reported results. Procedures for the issue of amended or revised results shall ensure that the reason for the change is recorded and, where relevant, included in the revised report. Revised results are delivered only in the form of an additional document or data transfer, clearly identified as a revision, and include the date and patient identity from the original report. The user is notified of the revision. When it is necessary to issue a completely new report, it is uniquely identified and contains a reference and traceability to the original report it replaces. Where the reporting system cannot capture revisions, a separate record of such revisions is maintained. Post-examination handling of samples. The laboratory shall establish and document the duration for which samples are retained after examination, as well as the storage conditions to be applied. After completion of the examination, the laboratory shall ensure that patient and source identification of the sample are maintained at all times. The suitability of the sample for any additional examination is known and recorded. The sample is stored under conditions that optimally preserve its suitability for further testing. The sample can be easily located and retrieved when required, and the sample is ultimately discarded in a manner that is safe, compliant with regulations, and appropriate to the sample type. Non-conforming work. The laboratory shall establish and maintain a documented process for managing situations where any aspect of laboratory activities or examination results fails to conform to its own procedures, define quality specifications, or user requirements, for example, equipment or environmental conditions outside specified limits, or monitoring results not meeting required criteria. The process shall ensure that responsibilities and authorities for the management of non-conforming work are clearly defined and documented. Immediate and long-term actions are determined and implemented, based on a risk analysis process established by the laboratory, to minimize patient safety risks and prevent recurrence. Examinations are halted and reports withheld whenever there is a risk of harm to patients. An evaluation of the clinical significance of the non-conforming work is conducted, including an impact analysis on examination results that were, or could have been, released prior to the identification of the non-conformance. A decision on the acceptability of the non-conforming work is made and documented. Where necessary, examination results are revised and users notified promptly of any changes that could affect patient care. The responsibility for authorizing the resumption of work is clearly specified. The laboratory shall implement corrective actions that are commensurate with the risk of recurrence of the non-conforming work. The laboratory shall retain records of non-conforming work and actions taken, as specified. Control of data and information management. The laboratory must have access to all the data and information required to carry out its work effectively. The term laboratory information systems refers to the management of information in both computerized and non-computerized systems. While many of the requirements relate more directly to digital systems, they still apply, where relevant, to manual processes as well. Risks associated with computerized laboratory information systems are outlined in ISO 22367, which provides guidance on identifying and managing potential vulnerabilities that could affect patient care or data accuracy. To ensure that information remains confidential, accurate, and available when needed, laboratories can rely on the information security controls and best practices described in ISO IEC 27001. These standards help establish strong safeguards to protect sensitive laboratory data throughout its life cycle. Authorities and responsibilities for information management. The laboratory shall ensure that the authorities and responsibilities for the management of information systems are clearly defined and documented. This shall include, but not be limited to, the maintenance, modification, and oversight of information systems in circumstances where such activities have the potential to impact patient care. The laboratory shall retain ultimate accountability for the performance, integrity, and reliability of its information systems, irrespective of whether activities are performed internally or by external providers. Information systems management. The systems used for the collection, processing, recording, reporting, storage, or retrieval of examination data and information shall comply with the following requirements. Each system shall be validated by the supplier and subsequently verified by the laboratory for functionality prior to introduction into use. Any changes to the system, including software configuration within the laboratory or modifications to commercial off-the-shelf software, shall be authorized, documented, and validated prior to implementation. Note 1. Validation and verification shall include, where applicable, the proper functioning of interfaces between the Laboratory Information System, LIS, and other relevant systems such as laboratory equipment, hospital patient administration systems, and systems in primary care. Note 2. Commercial off-the-shelf software, when used within its designed application range, may be considered sufficiently validated, for example, word processing, spreadsheet, or quality management software. Documented, with documentation readily available to authorized users. Documentation shall include information necessary for the day-to-day functioning of the system. Implemented with cybersecurity safeguards to protect against unauthorized access, tampering, and data loss. Operated in compliance with supplier specifications. For non-computerized systems, operation shall ensure that the accuracy of manual recording and transcription is safeguarded. Maintained to preserve the integrity of data and information, including recording of system failures, implementation of appropriate immediate and corrective actions, systematic and appropriate checks of calculations and data transfers. Downtime plans. The laboratory must have a plan in place to ensure that its work can continue even if an information system fails or goes offline. This includes being prepared for interruptions that affect automated processes such as selecting results, validating them, or generating reports. In the event of system downtime, the laboratory should be able to switch to predefined alternative procedures, so that testing, result review, and reporting can still be carried out safely and accurately, until normal operations are restored. Off-site management. When the laboratory information systems are managed and maintained off-site or by an external provider, the laboratory shall ensure that the provider or operator of the system complies with all applicable requirements of this document. Responsibilities, authorities, and service expectations are clearly defined in formal agreements or contracts. Compliance with requirements is regularly reviewed and verified by the laboratory. Patient data confidentiality, integrity, and availability are safeguarded through appropriate information security measures. Complaints. Handling of complaints. The laboratory shall establish and implement a process for handling complaints. This process shall include at least the following: a clear description of how complaints are received, substantiated, and investigated, and how decisions are made regarding the actions to be taken in response. Note: the resolution of complaints may result in corrective actions or serve as valuable input into the continual improvement process. A system for tracking and recording all complaints, along with the actions undertaken to resolve them. Assurance that appropriate actions are taken in response to each complaint. Furthermore, a description of the complaint handling process shall be publicly available to ensure transparency. Receipt of complaints. Upon receipt of a complaint, the laboratory shall confirm responsibility, determine whether the complaint relates to laboratory activities for which the laboratory is accountable. If confirmed, the laboratory shall proceed to resolve the complaint. Gather information, The laboratory receiving the complaint shall be responsible for collecting all necessary information to assess whether the complaint is substantiated. Acknowledge and communicate. Whenever possible, the laboratory shall acknowledge receipt of the complaint and keep the complainant informed by providing the outcome and, if applicable, interim progress reports. Resolution of complaints. The investigation and resolution of complaints shall be conducted in a manner that ensures fairness and impartiality. Under no circumstances shall the process result in discriminatory actions against the complainant or any party involved. Resolutions shall be made by, or at minimum reviewed and approved by, individuals who were not directly involved in the subject of the complaint. In cases where resources do not allow this separation, the laboratory shall adopt an alternative approach that safeguards impartiality and prevents bias in the resolution process. Continuity and emergency preparedness planning. The laboratory shall identify risks associated with emergency situations or other conditions in which laboratory activities may be limited or unavailable. To ensure resilience, a coordinated strategy shall be in place that includes plans, procedures, and technical measures designed to support the continuation of operations following a disruption. These plans shall be periodically tested, and the laboratory's response capability exercised whenever practicable. As part of this preparedness, the laboratory shall establish a planned response to emergency situations, considering the needs and capabilities of all relevant laboratory personnel. The laboratory shall provide information and training, as appropriate, to relevant laboratory personnel. Respond effectively to actual emergency situations. Take action to prevent or mitigate the consequences of emergency situations, in proportion to the magnitude of the emergency and its potential impact.